Assalamualaikum and good morning. I bid to Dr. Nurul and my fellow friends. We are from Group 3 and today we will be presenting our case study entitled Development of Covid-19 Detection Kit. The detection kit that we are going to present is loop-mediated isothermal amplification, or also known as LAMP. So this is our team members. The first one is me, Nurul Harizah binti Abu Hassan 198983. And then we got Nurul Izzah binti Rosli 198714, Shatirah Sofi binti Saifudin 197294, Wan Nur Adilla binti Wan Mustafa Kamal 197513, and lastly Wan Muhamad Zaim Zikri bin Zaiudin 196670. So this is our table of content. We will be presenting about the purpose, principle, methodology, advantages and limitation of LAMP, as well as method for improvement. So now let's move on to the purpose of LAMP. Recently, a novel coronavirus disease 2019 has emerged, rapidly spreading and severely straining the capacity of global health community. As you can see from the slide, in the mid of June 2021, a total number of 178 million COVID-19 cases with 3.85 million of deaths have been reported worldwide. Moreover, in Malaysia, 685,000 COVID-19 cases with 4.2 of death has also been recorded in the mid of June. So, the widespread of COVID-19 have urged many nations to employ a combination of containment and mitigation strategy to control and reduce COVID-19 cases. The combination of containment and mitigation strategy include social distancing, swab tests, working from home, country lockdowns, and development of various types of vaccines. According to the Ministry of Health Malaysia, early detection is also vital in controlling illness progression and limiting virus spread within the populations. Thus, a rapid and accurate method of early detection are vital to contain COVID-19 and prevent further spread, as well as predict subsequent infection waves of viral recurrence in the future. So LAMP is one of the methods to control widespread of COVID-19. LAMP assay is a nucleic acid amplification technique that is primarily used for diagnosis and detection of COVID-19. Generally, LAMP assay is used to amplify, detect, and identify specific virus, foodborne pathogen, disease, and food allergy. In the case of COVID-19, LAMP assay is purposely employed for amplification, where we amplify the target gene in RNA of COVID-19. We also use LAMP assay for rapid detection and identification of RNA from COVID-19 in human body, so that we can know whether a person has been infected with COVID-19 or not. So this is all for me. I will pass the presentation to the next presenter.
My name is Wan Nur Adilla binti Wan Mustafa Kamal and my matric number is 197513. Next, I will be presenting about the principle of LAMP. LAMP is the amplification and detection of the nucleic acid which could be completed at the same time. So we call it as a single-step assay. So, it involves a strand displacing DNA polymerase, which is employed to initiate the synthesis, while two primers from loop structures are used to facilitate and accelerate subsequent rounds of amplification. The LAMP process involves the incubation of nucleic acid sample, 4 or 6 specifically designed primers, and BST DNA polymerase in the same tube. Then it utilized 6 different target sequences simultaneously identified by separate distinct primers in the same reactions. So the functions of BST DNA polymerase is used to employ to initiate the synthesis. It is carried out in an isothermal setting, temperature of 60 to 65 degrees Celsius. So the changes in the temperature is not necessary. As mentioned earlier, LAMP assay involves 6 types of primer. They include 2 inner primers, which are the forward inner primer and backward inner primer. The other 2 are the outer primers, which are the forward outer primer and backward outer primer. So the additional primers that are involved are the two loop primers, which are the forward loop primer and backward loop primers. The function of the two loop primers in the LAMP assay is used to accelerate amplification and detection efficiency. So this is the sequence of COVID-19 primers that we will be using in order to detect the COVID-19 using LAMP assay. The working principle of LAMP assay involves three steps, which are the initiation, the cycling amplification, and the last one is the elongation process. For the first step, which is initiation, the F5P primer will hybridize to the F2C in the target DNA and initiates the complementary strand synthesis using a polymerase with displacement activity, which is the BST polymerase. Next, the outer primer of F3, which is a few bases shorter and lower in concentration than F5P, will slowly hybridize to the F3C in the target DNA and initiates the strand displacement DNA synthesis, releasing a FIP link complementary strand which can form a loop-out structure at one end. So this single-stranded DNA will serve as template for the BIP-initiated DNA synthesis and subsequent the B3' strand displacement DNA synthesis, leading to the production of the dumbbell-like shape of DNA. The second step is the cycling amplification. The FIP primer will hybridize to the loop in the stem loop DNA and prime strands displacement DNA synthesis, generating as an intermediate one-gap stem loop DNA with an additional inverted copy of the target sequence in the stem and a loop formed at the opposite end via the BIP sequence. The last one is the elongation process. The elongation and cycling steps occur with exponential amplification of the template with a dumbbell-like structure. So at the end of the cycle, LAMP products with cauliflower-like structures are formed with inverted repeats of the target sequence on the same strand.
Thank you. Next, I will continue the presentation for the third part, which is the methodology of LAMP, which is loop-mediated isothermal amplification. Before that, my name is Wan Mustafa Kamal Zikri with method number 196670. Okay, for this methodology, we will use the method that have been applied in one kit that is established by Aken Chemical Co. Ltd. And this is one of the company that involved in infection diseases. This company also involved in manufacturing and sales of clinical diagnostic and equipment. One of them is Lupam SARS-CoV-2 detection kit. They apply the LAMP principle. The methodology of this detection can be simplified into 6 steps. Start with sampling, preparation of reagent, preparation of sample solution, reagent and sample mixing, amplification by using incubator and thermal cycler, and the last one is interpretation of the result. Before start the process, we will prepare the materials and apparatus needed. We will need micropipette and tips, aluminium rack, simple centrifuge for 8 microtubes, incubator with hot burnet, heating block, UV light with 2 wavelength, and goggles and a protective eye mask. For extraction step, we can use Lupam viral RNA extraction kit that can be obtained from the same company. And the main kit is Lupam SARS-CoV-2 detection kit that contains dry RNA amplification reagent, Privomid and CV19 positive control and CV19 and negative control. Okay, the first step is sampling. Sampling will involve the swab technique. The specimen will be taken from upper respiratory tract, which is nasopharyngeal, and lower respiratory tract, which is bottom. The second step is preparation of reagent. First, dry RNA amplification reagent. This reagent will be stored in refrigerator, which is from 2 until 8 degrees Celsius for 5 minutes. After 5 minutes, take out the required number of tubes from the reagent based on the number of samples plus 2. and stored on ice. The remaining reagent will be kept in the original aluminum pack and placed back in the refrigerator. For the primer mist and positive and negative control will be centrifuged before use. After that, we go to the third, which is preparation of sample solution. The sample will be extracted using low pump viral RNA kit to obtain the RNA solution as the sample solution. They will take 5 minutes. Next is reagent and sample mixing. 5 microliter of primer mist will dispense into reagent tube with 10 microliter of sample solution to make a total volume of 35 microliter and close the cap. In other two tubes, 10 microliter of negative control and positive control are dispensed and close the cap. Spin down the tubes using a centrifuge. Then the dry reagent in the cap is reconstituted by inverting the reagent tube and collecting the solution in cap. The tube are left standing upside down for 2 minutes for 5 times to reconstitute the reagent until all the reagent are fully dissolved. After that, spin down again using centrifuge. Next step is amplification. This step will involve the use of incubator and thermocycler for visual fluorescence detection technique. The temperature of the incubator is set at 62.5 degrees Celsius. Then the reaction tube is slowed for 35 minutes for the amplification. After 35 minutes, the reaction will be inactivated using heating block at 95 degrees Celsius for 2 minutes. The last step is interpretation of result using UV light. Irradiate the bottom of each tube from side through goggles or any protective equipment. Before evaluate the sample, the run must undergo validity. For validity of the run, the positive control will emit green fluorescence light, while the negative control will not emit fluorescence light. After confirming the run is valid, the sample are evaluated. Positive sample will emit a green fluorescence light and negative sample will not. So, the duration for this detection test is 58 minutes. Preparation of dry RNA amplification reagent for 5 minutes. Preparation of sample solution is 5 minutes. Preparation of reagent is 10 minutes. Amplification reaction is 35 minutes and 2 minutes for inhibiting the reaction. I think that's all for me. Next, I will pass to the next person. Thank you.
Hi, Assalamualaikum. My name is Nurul Izzah binti Rosli. My metric number is 18014. So now, let's look at the advantages and limitations of LAN. The first advantage is that LAN is a quick technique, as LAN is an ultra-sensitive nucleic acid amplification method which capable of detecting many amounts of DNA or RNA templates in less than an hour. Besides, LAN method also is simple and cheap. This method can be performed in hospital laboratories and doesn't require expensive reagents or instruments, which reduce the time between taking a sample and receiving a diagnosis. The next advantage of LAN is that LAN is highly sensitive and specific. 1 to 10 copies of viral RNA template per reaction were sufficient for detection, which was 100-fold more sensitive than traditional RT-PCR methods. The next one is that LAN is flexible, as the AC can accept unprocessed samples, meaning that LAN method doesn't require any RNA isolation before performing the AC, and the results also can be read visually or by our naked eye. LAN method also has several limitations. The first one is that LAN is lack of research background. This is because the technology is newer than RT-PCR and doesn't have as much research behind it. So, Covid-19 tests using LAN technology are still being implemented in clinical settings. Next is complexity. The science involved in developing this test is more difficult than RT-PCR. This is because LAN method requires a complex parallel design system that can limit target site selection, resolution, or specificity. Next one is that LAN has limited detection, meaning that LAN tests for COVID-19 can only determine whether or not a person is currently infected with coronavirus. It cannot provide information on other diseases or symptoms, and it also cannot tell us if a patient has previously been infected with the virus or if the patient is immune to the virus. The last one is that LAMP has a high risk carryover contamination. The high risk of carryover contamination caused by opening post-LAMP vessels and using sensors, which poses a significant risk to reliable results, meaning that carryover contamination can lead to a positive result due to the extremely high efficiency of LAMP relation. So that's all from me. Now I will pass to the next member. Thank you.
Assalamualaikum and greetings to my friends and to Dr. Nurul. My name is Shatirah Sofi binti Saifudin, and I'll be taking over to explain about the methods of improvement for loop-mediated isothermal amplification, or also known as LAMP assay. So firstly, LAMP assay can be improved by the addition of recombinant A combined with the betaine. So this combination able to reduce the threshold time by 3 minutes. Other than that, LAMP assay can also be improved by the combination of recombinant A with ATP that has been proven to reduce the threshold time by 7 minutes. However, according to a study by Zaim in 2020, the addition of ATP alone can greatly improve LAMP assay by reducing the threshold time by 6 minutes. Next, LAMP can also be enhanced by the addition of nucleotides that is other than ATP. The addition of nucleotides help to make the reactions become more faster, so lead to reduction in reaction time. Other than that, LAMP assay can also be improved by the addition of new sets of primer, or also known as swarm primers. So the term of swarm primers are based on their relatively high concentration and their ability to create new amplicons, although swarm primers are known in lacking of single-stranded annealing sites. The addition of swarm primers in LAMP assay able to increase the amplification speed, increase the indicator contrast, and increase the reactions product. Lastly, LAMP can be improved by including betaine in the assay. This is because exclusion of betaine will increase the risk of producing false positives result, which we want to avoid. Based on the studies by Zhou et al. In 2020, the addition of betaine into LAMP assay does not produce any false positives result at all, and the inclusion of betaine help to avoid unspecific amplifications in this assay. So I think that is all from me and my group mates. Thank you.